Welcome to another deep dive. Today we're going to be looking into systems biology, and we're going to be exploring some fascinating ideas about how life really works. You're interested in biological complexity, and we've got a great stack of sources for this deep dive, from Dennis Noble's The Music of Life to some really thought-provoking academic articles.
Yeah, it's exciting to delve into this field.
So get ready to challenge some of those classic assumptions about DNA, genes, and maybe even evolution itself.
Absolutely. It really pushes us to think beyond the traditional gene-centric view of biology.
Right, like we've all heard that DNA is the book of life, but is it really that simple?
Right.
And what about the idea of selfish genes? Do genes actually act like little agents out for their own good?
Those are great questions, and systems biology suggests that things are much more nuanced than that.
Right.
It encourages us to look at life as a complex web of interactions where different levels of organization, genes, proteins, cells, organs, and the environment are all influencing each other.
Okay, I'm ready to have my mind blown. Let's start with something I vaguely remember from biology class, the central dogma of molecular biology.
Okay.
It's this idea that information flows in one direction, from DNA to RNA to proteins. That always seems so straightforward.
It's a powerful concept, but even fundamental principles can be challenged as we learn more.
Right.
One of the sources highlights a discovery that really shook things up: reverse transcriptase.
Wait, doesn't that have something to do with retroviruses? Like how HIV inserts its genetic material into our cells?
Exactly. Retroviruses use reverse transcriptase to do something pretty remarkable.
Okay.
They take their RNA and actually use it as a template to create DNA, which then gets integrated into the host cell's genome. That's completely flipping the script on the whole DNA to RNA flow of information.
So the central dogma wasn't quite as dogmatic as we thought.
Yeah.
And this isn't just some rare exception, right? Retrotransposons, which are similar to retroviruses, are found throughout many genomes, including our own.
Right. They make up a significant portion of our DNA, and their ability to move around and copy themselves using reverse transcriptase has had a huge impact on genome evolution.
Wow.
This brings us to a really important point. Maybe we need to rethink our view of the genome as a static blueprint.
Instead of thinking of it as a rigid set of instructions, what if we saw it more like a dynamic database?
That's a really powerful analogy. The genome is like a vast library of information that the cell can access and utilize in different ways depending on the context.
Okay.
It's not just about the genes themselves, but how they're regulated, expressed, and interact with each other and the environment.
So it's less about a predetermined program and more about a flexible system that can respond to different situations. Are there any good examples of how this actually plays out in living organisms?
Our sources use the example of circadian rhythms, those internal clocks that regulate our sleep-wake cycles and so many other physiological processes.
Okay.
It turns out that the 24-hour cycle isn't just dictated by a single gene,
Right.
but rather emerges from a complex interplay of multiple factors.
So it's not just the DNA calling the shots, it's the whole system working together.
Exactly. You have the period gene, which codes for the PER protein, but you also have the structure of the cell itself and environmental cues like light and darkness all interacting to create that rhythmic cycle.
Okay.
If you remove any part of that network, not just the gene, but any part, the rhythm gets disrupted. This suggests that control is distributed throughout the system, not localized in a single gene.
That's a much more dynamic picture than just a gene dictating a trait. This makes me wonder about those gene knockout experiments scientists do. If a single gene isn't always the sole determinant of a trait, does knocking it out really tell us the whole story?
That's a great question, and it leads us to another key concept in systems biology: genetic buffering.
Okay, let's unpack that. What is genetic buffering and why does it matter?
Organisms are remarkably resilient, and one of the ways they achieve this resilience is through genetic buffering. Essentially, they have backup mechanisms that can compensate for the loss or malfunction of certain genes.
So even if you knock out a gene, the organism might have a plan B or even a plan C to keep things running smoothly.
Precisely. This has huge implications for how we interpret gene knockout studies.
Okay.
Our sources point out what they call the genetic differential effect problem.
Okay.
If we only focus on the differences caused by removing a gene, we might miss the bigger picture of how that gene normally functions within the context of the whole system.
That's fascinating. So how do we get a more complete understanding of gene function if simply knocking it out doesn't always give us the full picture?
One powerful tool that systems biology offers is reverse engineering.
Okay.
Instead of just breaking things apart, we can try to build them up. By modeling biological systems at different levels of organization, from cells to organs to whole organisms, we can start to see how all the pieces fit together and how genes contribute to those higher level processes.
It's like taking apart a clock to see how all the gears and springs work together. But instead of a clock, we're trying to understand something as complex as a beating heart or a developing brain.
Exactly. And one of our sources gives a great example of how this works in practice. Researchers created a model of the heart's rhythm and found that reducing the activity of a specific ion channel, which is like a tiny gate that controls the flow of charged particles, had a surprisingly small effect on the heart rate.
Why was that? Did the heart have some kind of workaround?
Precisely. The model revealed that the heart had compensatory mechanisms that kicked in when that particular ion channel's activity was reduced.
Huh.
These mechanisms allowed the heart to maintain its rhythm even with that change.
Wow.
This is something that traditional gene knockout experiments might have missed.
That's incredible.
Yeah.
So the model allowed them to see the bigger picture and understand how that gene actually functioned within the context of the whole system.
Exactly. And this brings us to one of the most thought-provoking ideas in systems biology, Dennis Noble's theory of biological relativity.
Biological relativity. Now we're getting into Einstein territory. What does that even mean?
It's a fascinating concept. Noble argues that there is no single privileged level of causation in biological systems. It's not just about genes dictating everything from the top down. Higher levels of organization, like cells, tissues, and organs, can also influence gene expression and function.
So instead of a one-way street from DNA to organism, it's more like a two-way street with constant communication between different levels of organization.
That's a great way to put it. Think about the heart pacemaker cells.
Okay.
The rhythm itself doesn't exist at the molecular level. It's an emergent property of the cellular system.
So you're saying that the individual molecules don't contain the blueprint for the rhythm that emerges from their interactions within the cell.
Exactly. And this has profound implications for how we understand biological complexity. Just as you can't fully understand a symphony by analyzing a single note, you can't fully understand life by focusing solely on genes.
Right.
We need to consider the context, the interactions, the emergent properties that arise from the symphony of biological systems.
Wow, this is really making me rethink some of those old assumptions about how life works. But before we dive any deeper into the implications of this new perspective, let's take a moment to reflect on what we've learned so far. We'll be back in a flash to continue our exploration of systems biology and its fascinating challenges to traditional biological thinking.
You know, we were talking about the central dogma and how even fundamental principles can be challenged as we learn more.
Aye.
Makes you wonder about other widely accepted ideas in biology, like the concept of the selfish gene.
Yeah, that's a really kitschy phrase. But now I'm wondering if it's an accurate way to describe how genes actually function within these complex systems we're discussing.
It's a powerful metaphor, and it has been incredibly influential in evolutionary biology. The idea is that genes are essentially competing for survival, and that their selfish drive to replicate is what drives evolution.
But doesn't that kind of anthropomorphize genes a bit too much?
Right.
We're not talking about conscious agents with desires, are we?
All right, that's a valid point. Genes as DNA sequences aren't actually capable of selfishness. They're acted upon by the cellular environment, which includes all sorts of things like proteins, membranes, and signaling molecules.
Right.
It's this dynamic interplay of all these elements that drives biological processes, not some inherent selfishness of the DNA itself.
So maybe the focus on individual genes being selfish is a bit misleading. It's more about the whole system and how different components interact and influence each other.
Exactly. And this highlights another important point that often gets overlooked when we focus solely on genes. Inheritance isn't limited to DNA. Cells pass on a wealth of information beyond the nuclear genome.
You mean like the mitochondria? I remember learning that they have their own DNA.
Yes, mitochondria are a prime example. They're thought to have originated from ancient bacteria that were engulfed by other cells, and they still retain their own DNA, which codes for some of their essential functions.
So we inherit mitochondrial DNA from our mothers, right?
That's right. And it's not just mitochondria. Plant cells have chloroplasts, which are also thought to have originated from ancient bacteria and have their own DNA. And there are other organelles and cellular structures that play a role in inheritance beyond the nuclear genome.
I'm curious, how does this cellular inheritance play into our understanding of evolutionary processes?
Well, it suggests that evolution isn't solely driven by changes in the DNA sequence. The inheritance of organelles, cellular structures, and even certain patterns of gene regulation can all contribute to evolutionary change.
So we're talking about a much broader and more dynamic view of inheritance than just the passing down of genes from parents to offspring.
Precisely. And one of our sources highlights a really interesting phenomenon that challenges the traditional view of inheritance epigenetic modifications.
I've heard that term before, but I'm not quite sure what it means. What are epigenetic modifications and why are they important?
Think of epigenetic modifications as switches that can turn genes on or off without actually changing the DNA sequence itself. These switches can be influenced by a variety of factors, including environmental cues like diet, stress, or exposure to toxins.
So the environment can actually affect how our genes are expressed.
Exactly. And what's really fascinating is that some of these epigenetic changes can be passed down to future generations.
Wow.
This means that the experiences of your parents or even grandparents could potentially influence your own gene expression.
Wow, that's both incredible and a little bit unsettling. It means our genes aren't our destiny, right?
It certainly adds another layer of complexity to our understanding of inheritance. It
Hmm.
means that our genes are constantly interacting with our environment and that this interplay can have lasting effects.
So are you saying that Lamarck might have been onto something with his idea of the inheritance of acquired characteristics? I thought that had been
That's
completely debunked.
It's more nuanced than that. Lamarck's original idea was that organisms could pass on traits they acquired during their lifetime directly to their offspring, like a giraffe stretching its neck to reach higher leaves. While that's not quite how it works, epigenetic inheritance does show that the environment can influence heritable traits in a way that Lamarck might have found intriguing.
This is really challenging the classic view of evolution as a slow, gradual process driven solely by random mutations.
It is. And one of our sources even mentions the work of Conrad Waddington, a developmental biologist who conducted experiments in the mid-20th century that showed how environmental stress could induce new phenotypes in organisms which could eventually become heritable. He called this process genetic assimilation.
So the environment isn't just a passive backdrop for evolution. It can actually shape the genetic makeup of populations over time. That has huge implications for how we think about the evolutionary process.
It certainly does. It suggests that evolution is much more dynamic and interactive than we previously thought. It's not just about random mutations being selected for or against. It's also about the environment influencing which genes are expressed and how those patterns of expression can be inherited.
This is all so fascinating,
Hmm.
but I have to admit it's a lot to wrap my head around. It seems like we're moving beyond the traditional framework of the modern synthesis of evolutionary theory, which emphasizes random mutations and gradual change.
You're right, the modern synthesis has been incredibly influential, but as we learn more about these complex interactions between genes, organisms, and their environment, it's clear that we need a more expansive framework to encompass all these new discoveries.
So where do we go from here? If the modern synthesis is no longer sufficient, what new models are emerging to explain this more dynamic and interactive view of evolution?
Well, some researchers are proposing what they call an integrative synthesis of evolutionary theory. It aims to incorporate all these new findings about things like epigenetic inheritance, symbiogenesis, horizontal gene transfer, and the role of development and plasticity in evolution.
It sounds like we're moving towards a more holistic and nuanced understanding of evolution, one that recognizes the interplay between physiological function, environmental influences, and genetic change.
Exactly. And this is where systems biology really shines because it provides the tools and the conceptual framework to study these complex interactions and unravel the mysteries of life in all its dynamic glory.
Speaking of unraveling mysteries, we've covered a lot of ground in this deep dive. It's time to bring it all together and reflect on the key takeaways and the implications of this new understanding of life. Stay tuned for our final segment where we'll wrap things up and leave you with some thought-provoking questions to ponder. We've journeyed through some pretty mind-bending concepts in this deep dive into systems biology and biological complexity. It's amazing how much our understanding of life has evolved beyond that simple idea of DNA as the book of life.
Absolutely. We've seen how genes are not isolated actors, but rather participants in a complex and dynamic web of interactions. Their activity is influenced by the cellular environment, by epigenetic modifications, and even by the experiences of previous generations.
It's like realizing that a single musical note doesn't tell you much about the symphony. It's the interplay of all the instruments, the rhythm, the harmonies that create the beauty and complexity of the music.
That's a wonderful analogy. And just as a conductor shapes the performance of an orchestra, the cellular environment orchestrates the expression of genes, shaping the development and function of the organism.
So if the book of life metaphor is inadequate, what new metaphors might capture this more dynamic and interconnected view of life?
That's a great question, and it's one that scientists and philosophers are grappling with as we move beyond the traditional reductionist view of biology. Dennis Noble, whose work we've discussed, proposes that music itself offers a more apt analogy.
I love that idea. It evokes a sense of fluidity, of emergent properties of interconnectedness that seems to resonate with what we're learning about biological systems.
And it emphasizes the importance of understanding life as a process, a continuous dance of interactions, rather than a static blueprint.
So what does this all mean for you, dear listener? As you navigate your own life, consider the implications of systems biology for understanding your health and well-being.
Remember that your genes are just one part of a much larger story. Your lifestyle choices, your environment, even the experiences of your ancestors all contribute to the intricate symphony that makes you you.
And as you ponder the mysteries of life, perhaps you'll find yourself listening for those subtle harmonies, those emergent properties that make life so much more than the sum of its parts. Keep exploring, keep questioning, and who knows what amazing discoveries await you in this grand symphony of life.